Hello guys, and welcome to yet another video of chemistry for Wroclaw Medical University students, and for anyone who's really interested about biochemistry, really. So today we have quite a long topic. Today's topic is peptides, and yeah, so basically first we're gonna cover like the structure of amino acids, stereochemistry, and then you guys are going to have to learn the essential and non-essential amino acids and all the amino acids with their different classifications. And then we're going to dive into the chemical reactions of amino acids and stuff like that. So to start off with, what is an amino acid? So first of all, an amino acid is the building block of a protein. And like we know, proteins are required for a lot of different biological activities, such as making enzymes, and you have also fibrous proteins, which make up stuff like the skin, the cartilage, and so on. So first of all, the general structure of amino acids, the amino acid that we're going to be focusing on is alpha amino acids, because these are the ones that are usually present in biological tissue. An alpha amino acid has two functional groups, a carboxylic acid and an amino group, hence amino, because you have an amine group, NH2, and then you have a carboxylic acid group, which is this one here, C-double-bond-O-OH. And as we know, carboxylic acid, there's an acid in there, so we know that this can act as an acid by donating a proton. And this is the Brown-Stidd-Lowry definition of an acid, if you remember when we were talking about acid and bases. And then we also have NH2 here, okay? An amino group. An amino group can also act as a base because it can receive a proton, okay? So you have what's called an ampholyte, okay? So you have something that can act as an acid by donating its proton, but also as a base by accepting a proton, okay? And then you're going to have the variable part. So the variable part, or the side chain, is characteristic for the amino acid, a certain amino acid that you have. So it would be different for different amino acids. Like, for example, for alanine, it would be a methyl group, like you see here. Like you see here, that's alanine. You have the methyl group, okay? And you get more complex side chains, like, for example, let's say histidine. Here you have quite a lot of NH, okay, NH plus. So yeah, this is the variable group, the side chain, okay. This group depends on which amino acid is in question, okay, and it can be multiple groups. It can be can contain sulfide, can contain thiols, which contain sulfur, and it can contain alkyl groups, so that's CH3 or CH2, and so on. So or it can be aromatic, so it can contain a phenyl group, like phenylalanine, for example. Okay, so one thing to note about amino acids, okay, is that if you notice, apart from one amino acid, which is, which one was it? The one with the side chain of H, glycine. All amino acids have chiral carbons, okay, which is very interesting. What does a chiral carbon mean? So this is where stereochemistry comes in. So first of all, we're all used to seeing the 4 bonds around the carbon this way. However, in nature, or usually it's not like this. There's a geometric arrangement, which I will try to edit into this video, but again, my skills I don't know if they will be enough for that. But yeah, you have 4 different groups around a central carbon, okay? So that means that you can get 2 different arrangements, the L arrangement and the D arrangement. So these are called enantiomers, and they're basically mirror images of each other, the same as your hands. So if you put your hands together, oh, it's gonna be hard. Okay, if you put your hands together like this, you will see that they're pretty much the same, but they're mirror images of each other. What does that mean? Is that they're non-superimposable. They don't go perfectly over each other. You see the thumb sticking out, you know, and the order is wrong because they're mirror images of each other. Same as when you look in a mirror and you see your left is right and your right is left. Okay. All right. So that's what chirality means, and enantiomers and L and D configurations pretty much is that they have for the L configuration you will see that the carboxylic acid group and the amino group are on the left side, okay, whereas here they're on the right side, okay, and that's L and D. Okay, so now that we've gotten that out of the way, we can start looking at individual amino acids. So first of all, amino acids can be grouped into first of all, non-polar amino acids, polar amino acids, acidic amino acids, and basic amino acids. So first of all, polar and non-polar. What's the difference between polar and non-polar? From the name, polar meaning it has two poles, okay? And since it has two poles, that means it has a positive pole and a negative pole, or a slightly positive and slightly negative, therefore making it soluble in water, okay? Whereas non-polar tends to be insoluble in water because it doesn't have the polar nature that polar amino acids have, okay? So it will be an alkyl or even a structure that resembles a fatty acid, you know, so it will have a lot of CH3 and CH2, but it won't be polar because there are no two poles of the side chain, okay? And then you have acidic amino acids, and as we agreed several times before, acidic meaning that it has a tendency to donate a proton, okay? So you will see that these are the ones that have maybe a carboxylic acid group or a thiol or something like that, something that's able to give away a proton. And then you're going to have the basic ones, and these are the ones that have the side chains with nitrogen, okay? So it like they have a tendency to attract protons rather than donate protons. So as for the structure of the amino acids, you guys should have this in the book. It is beneficial to learn them. I would say learn out of each group the most three prominent ones for sure, but for the other ones, of course, like I'm just saying this because there's a lot of amino acids. There's 20 amino acids and different structures, especially for those who are not very acclimated with chemistry. They will find trouble trying to memorize all the structures. So I would say learn the most important amino acid out of each group. So learn maybe 4 out of the polar ones, 4 non-polar, and so on. 4 acidic, 4 basic. Okay. But yeah, if you really want to be safe, then of course learn them all and learn the classifications, you know. But the classifications if you know the structures, you can figure out the classifications, okay? Because you can see here you have an OH group, that's polar, isn't it? Okay, here you have an alkyl group, so that's non-polar. Okay, and it's easy, just as easy as that, to be honest. And you have here NH3, so that's basic. Okay, just from figuring out the structure, you can figure out the classification of an amino acid. So now we move on to amino acids as zwitterions and ampholytes. So ampholytes, the term that was mentioned at the start of the video, what does that mean? An ampholyte is a molecule that can act as both an acid and a base. So it can accept a proton, but it can also donate a proton. Okay. So yeah, what's a zwitterion? So zwitterion, there's an ion in there, so we know that it's going to be in an ionic form. However, in a zwitterion, you will see that the carboxylic acid part donates its proton, therefore becoming negatively charged. Okay, and the basic part accepts a proton, therefore becoming positively charged. And therefore, because this is negative and this is positive, and you will have also something happening in the side chain, whether it's acidic or basic, okay? And what you will see is that they become neutrally charged, so they don't have any positive or negative charge. They become neutrally charged, okay? And that's what zwitterion is that you basically have, you've lost the proton here. This is so to make carboxylate, and here you have a aminium, okay? Ammonium or aminium. No, it's aminium. Yeah, so you will see here the negative charge and the positive charge cancel each other out, and they form a neutral neutral molecule or neutral amino acid. So yeah, this zwitterion occur at very specific and very unique isoelectric points. So if you've watched the videos from chromatography and what was the other one? Electrophoresis, you will see that we separate them based on the isoelectric point. This is because the isoelectric point is unique. And what is the isoelectric point exactly? The isoelectric point is the pH at which the zwitterion occurs. Okay, so we said that the zwitterion is when you have the carboxylate, the carboxylic acid part becomes a carboxylate, and the NH2 or the amino part becomes an aminium. And the pH at which that happens, at which you get this neutrally charged amino acid, is different for each amino acid. Okay, and why is that? It's because there are different side chains or alkyl chains. Okay, I'm gonna try and sit down just a second. Oh my god. Okay. Yeah. So yeah, like you can see here, you will see that at certain pH you have this isoelectric point and it's neutrally charged. Below that pH, you will see that the base accepts a proton because it's more acidic than the isoelectric point. And above that pH, you will see that it donates a proton because the environment becomes more basic. Okay, and the molecule, the amino acid, wants to keep it stable. Okay, and then you can also, when you have different side chains, this is where stuff starts to get tricky. You can get different forms. Okay, so you can get a form of amino acid where the side chain has donated a proton, but the carboxylic acid hasn't, or vice versa. Okay, same concept with basic amino acids, and that's why acidic and basic amino acids tend to have high or low isoelectric points. Okay, because they have these side chains that also cause the isoelectric point to increase or decrease. Okay, so moving on, we have essential and non-essential amino acids. So these are you're just gonna have to know which ones are essential and which ones are non-essential. What does essential mean? Essential meaning that you cannot obtain it from, like, your body cannot synthesize this amino acid. Non-essential means that from your diet, from your usual diet, your body makes this amino acid on a daily basis, pretty much. So yeah, you have to obtain essential amino acids from your diet, whereas non-essential amino acids can be synthesized in the body. And you're going to learn in biochemistry later how amino acids are synthesized, so if you don't know already, don't worry about it. Okay, so now we have the peptide bond. So now we know that there's a part which contains a carboxylic acid and a part which contains an amino group. Okay, so what can happen with different amino acids is you can have a sort of chain of amino acids, which is also called a polypeptide because it's a polymer of amino acids being the monomer. Okay, so what if you don't know what polymer and monomer means? A polymer means a molecule that consists of repeating monomers. Okay, and a monomer is a certain molecule that repeats to form a polymer. Okay, so the peptide bond is, first of all, it's called an amide bond. Okay, and the peptide bond is formed by the reaction of the carboxylic acid of the carboxylate part reacting with the N terminal, or this is called the N terminal, and it's called the C terminal, by the way, of the adjacent amino acid. Okay, and then you get something called an amide bond. This is this structure highlighted here. This is the amide bond. Okay, and this is called the polypeptide bond when it happens between two amino acids. If it's inorganic, well, if it's non-medical chemistry, you would say just an amide. You wouldn't hear the word peptide a lot. So yeah, as opposed to isolated amino acids, peptide bond has different geometry. It's a plane. It's almost like a zigzag, you know, because by binding the carbon and nitrogen, and you also have the double bond oxygen, you're kind of flattening out the structure, okay, because of different interactions of the electrons. And this is where it starts to get interesting. So we all know that there's single bonds and there's double bonds, okay, in chemistry. However, the peptide bond is a different concept, because it's almost like a mixture of a single bond and a double bond, okay? And you can have the double bond either between the carbon and the oxygen, or the carbon and nitrogen, okay? So you get resonance, because bonds are not really fixed. They're probable areas where electrons might be. So sometimes you will see that electrons will form a double bond between the carbon and nitrogen, and sometimes they'll form the double bond between the carbon and the oxygen, okay? So you get this resonance. You get different sort of forms, okay? You'll see one where there's a carbon double bonded to a nitrogen, and one where it's double bonded to the oxygen, and it keeps switching, okay, until it reaches a stable structure. And yeah, you don't get this form a lot. It happens, but this is more preferred. But because you don't know what this is, or you might know what it is, but there's something called the electrochemical series, and oxygen and nitrogen are pretty close in terms of electronegativity, okay? So if you want to know more about this, you can research it. It's pretty interesting, but it's not required, so which is a shame. But yeah, now we go on to cis and trans configuration. So now we're talking about something called stereoisomerism. I don't know if you remember, but we did talk about this, I think, at the start of the beginning modules. But stereoisomerism, you have two forms. You have cis form and a trans form. Okay, and like we said, because this resembles a double bond, okay, the geometry changes. But double bonds, as opposed to single bonds, you cannot rotate double bonds around their own axis, okay? So their geometry doesn't allow you. So for a single bond, you can keep rotating around the central atom as much as you want. The angles are still going to be the same between each bond. You know, the geometry is still going to be the same, but you can rotate it around the central atom. Okay, and that gives it a unique sort of configuration, because it can sort of rotate. You can do that, but with a double bond, or in this case a semi-double bond, I just made that up, you will have a fixed bond, so you cannot rotate it around its axis. So you can either have the alpha carbon, which is the continuation of the amino acid, or the alpha carbon is this carbon. Hold on, it's this carbon, this central one. This is called the alpha carbon. Okay, so you will have the alpha carbon of the amino acid, of the two amino acids, either facing each other. So this is the opposite to each other, or in the same side. So if you have them both over here, so if you switch this around, you will see that this is the cis form. Okay, the carbon atoms are facing each other, but in this structure they're opposite to each other. You can see here. Okay, and that gives it a trans form. Okay, so that's the trans configuration of the peptide bond. Okay, and this is more dominant over the cis form, approximately 1,000 to 1. So that's quite a lot. But sometimes you will have, depending on the amino acids, you will have different ratios. So now we talk about N-terminal and C-terminal amino acid residues. So what's an N-terminal and what's a C-terminal? So first of all, we talked about the polypeptide chain. Okay, and you see the peptide bonds here. The N-terminal is the last nitrogen, okay, and it's a free amino group. Okay, this is basically, you have a chain, okay, and then you're gonna have one at the very start of it, you're gonna have an NH2. This is the N terminal, and this is the C terminal. Okay, and within the bonds themselves, you're going to have the N terminal and the C terminal, and so on. Okay, so now that we know what an N terminal and a C terminal is, we can use that in sequencing. So there are three methods. First of all, we talk about N terminal residue sequencing. Okay, so first of all, before you do any kind of sequencing, you need to eliminate something called disulfide bonds. So disulfide bonds, from the name, disulfide, so two sulfur sulfide. Okay, the disulfide bonds usually occur between two cysteine amino acids, okay, of different chains or the same chain. They basically make cross links between each other, and they help form the tertiary structure of a polypeptide or a protein. Okay, so you need to break these down in order to make it linear, in order to make the chain linear. Okay, and how you do that is by something called 2-mercaptoethanol or dithiothyretyl, okay, or DTT. Okay. So yeah, now that we've done that, now that we've eliminated all the disulfide bonds, we need to identify which is the N terminal and which is the C terminal. Okay, so the N terminal can be identified by sort of binding a reagent, okay, which labels the N terminal. Okay, and that what we can do next, it can also separate the N terminal after labeling it, and then we can see what the N terminal amino acid is, or what the amino acid that the last the first amino acid is, using chromatography. Okay, because now that you've labeled it, you can separate it, and then you can see what amino acid you have at the N terminal. Okay, so three methods that we're going to talk about is Sanger method, Dansyl chloride method, and Edman technique. Okay, so first of all, the Sanger method. So in the Sanger's method, we use something called Sanger's reagent, which is 2,4-dinitrofluorobenzene, or DNFP. Okay, just know the basic concept of it and what reagents you use for each method, and I think that will be enough for the test. So if it asks you about the Sanger method, you can say, oh, it uses DNFP, okay, and then that DNFP reacts with the N terminal residue, because that will, again, remember, that's what we're trying to label, or we're trying to see under alkaline conditions, and then after that it causes a hydrolysis of the N terminal amino acid, okay, and therefore it removes the N terminal amino acid from the chain. Okay, it removes the first amino acid from the chain, and then after that you will see that there's a new N terminal, and then it can bind to that N terminal, the DNFP. Okay, and then it will remove that one. It will hydrolyze the peptide bond with that one, and so on and so on and so on, until you can form separated amino acids, and then you can sequence them. You can tell which one was there first. Okay, and then you can have a sequence of amino acids, and you can even use that sequence to determine the genetic sequence, which is very interesting. Okay, so yeah, the Dansyl chloride is very similar, but in this case we're binding a fluorescent molecule, which is Dansyl chloride, and this Dansyl chloride reacts again with the N terminal amino acid to form the Dansyl amino acid, which is highly fluorescent. And because it's fluorescent, or it produces photons, we can detect these photons, okay, and detect the fluorescence, and therefore we can sequence them. Okay, so you'll see you will react with Dansyl chloride, and then you'll record the time. And then once you get the fluorescence, first you will hydrolyze it, and then you can separate it, and then you can see that they're fluorescent, or the amino acids are fluorescent at the points that they were separated. Okay, so it can also be used for sequencing. And then you have Edman's techniques, or Edman's degradation, and what you use for this is Edman's reagent, which is phenyl isothiocyanate, or PITC. Okay. And then under mildly alkaline conditions, so pH slightly above 7, it will react with the N-terminal to form a phenyl thiocarbonyl derivative of the polypeptide. So this sounds quite complicated, but phenyl thiocarbonyl is basically a derivative of, or the product of, the phenyl isocyanate reacting with the N-terminal amino acid. Okay, and then this causes a chain of reactions, which then causes the hydrolysis of the N-terminal, again, the N-terminal amino acids, which can then, now that we've hydrolyzed it and separated from the rest of the polypeptide chain, we can run it in chromatography and sequence the protein. Okay, and then you can do that again and again and again to find out the sequence of the protein. Okay, so yeah, you can also use exopeptidase. So these are enzymes that are specific to certain proteins, to certain amino acids, okay, or certain peptide bonds, and these will, if you add the required enzymes for each specific peptide bond for the two amino acids linked, you can find out the sequence, okay, by running it again through chromatography. Okay, so you're gonna have a lot of enzymes. It's gonna be very time costing and expensive because they're very specific. Okay, so now we have C-terminal residues. So like we said, the N-terminal is the free amino acid at the start, or the part where you have a free amino group. The N-terminal is the part where you have a free carboxylate group. Okay, so the method that we use in order to detect the amino acid at the C-terminal is called hydrazenolysis. Okay, and what happens is the first thing we use is something called hydrazine. Okay, at 90 degrees Celsius for from 20 to 100 hours under mildly acidic. So for the N-terminal, it's alkaline or mildly alkaline. For this one, it's mildly acidic. Okay, and this reaction, as you can imagine, the hydrazine reacts with a C-terminal amino acid to form something called amino acyl hydrazine. Okay, don't be intimidated. It just means it's a product of the first amino acid and the hydrazine. Okay, and then you will see that the C-terminal amino acid separates from the rest of the chain, so the peptide bond is hydrolyzed. Okay, and then you can use chromatography again to separate and identify the amino acid that you just separated. Okay. So now that we're done with C-terminal and N-terminal, this is quite a long topic. I don't know if I'm going to be able to fit it into one video. I'm going to try, but I don't think so. We're at 28 minutes already, so I'm just going to go on to biogenic. I'm going to finish up until biogenic amines, and then I'll leave the rest for the next video. So amino acid composition of proteins. First of all, in order to sequence proteins, you have, like we said, you can either do it through C-terminal analysis or N-terminal analysis, but you can also hydrolyze all the peptide bonds between all the amino acids, and then the free amino acids can be separated in acidic conditions, of course, and using temperature, and then you can also use some enzymes to separate these amino acids. Okay, but like we said, enzymes are highly specific for the peptide bonds that they hydrolyze, and after that you can use, guess what, chromatography. So disulfide bonds, we've already talked about this. Disulfide bridges or disulfide bonds happen between two cysteine, but they can also happen between other amino acids, but the main amino acid is cysteine. Okay, and you will see that the two cysteine molecules, because they have a thiol group, the sulfide ions make a bridge. Okay, and these bridges help to form cross links between cysteine, and the more cross links you have, the more structurally stable the chain is, or the structure, the overall structure is. And then you'll, as you expect, you can see quite a lot of that in proteins that require structural stability, or fibrous proteins. Okay, so in order to break down disulfide bonds, we already talked about this. Dithiotherol, or DTT, or 2-metacarboethanol, and yeah, that's how you hydrolyze the disulfide bridges. Okay, so natural peptides with biological activities. So now we're going on to specific peptides, or chains of amino acids that have function. Okay, so these can act as neurotransmitters, or they can act as messengers. Okay, and yeah, the most famous one is glutathione, or because it forms other stuff as well, like GSH and GSSG. You're going to learn more about this in biochemistry, but for now, just know which amino acids can act. Which peptides have biological activity and have biological function without needing to form proteins. And you can also have amino acids that can go modification in order to form messengers, second messengers, or they can form neurotransmitters, or they have also biological functions. And an example of that would be gamma aminobutyric acid, or GABA. Okay, and you're going to learn much more about that in physiology as well as biochemistry, and these are very important neurotransmitters. And you also have histamine. It's a derivative of the amino acid histidine. Okay, and it's a modulator of the inflammatory response. I'm running out of time here, so I'm trying to keep it short. And then we have the biogenic amines. So biogenic amines is basically a modified version of the amino acids that has an amine group. Okay, so an example of that would be histamine, serotonin, noradrenaline, epinephrine, dopamine, and ethanolamine. Okay, and L-dopa as well, and tyrosine, stuff like that. L-tyrosine, okay, can go a chain of reactions to form different biogenic amines, like L-adrenaline, noradrenaline, dopamine, and L-dopa. Okay, and polyamines they're another form of biogenic amines, and they're formed from ornithine. Ornithine is going to be important when you're learning about this transport of amino acids and synthesis of amino acids. Okay, and yeah, they also have biological function. So that's it for today. I'm going to talk later about the post-translational modification of amino acids. There's not much to know really, but it's going to take a small portion of the next video, which is going to be talking about also proteins, but I think in this module talking about the denaturation of proteins, and yeah, a type of proteins, the globular, and then they're also going to talk about proteins in colloidal solutions, and yeah, stuff like that. So this is the topic for next video. I'm also going to add to that the post-translational modifications, so acylation, carboxylation, acetylation, decarboxylation, or phosphorylation, all that kind of stuff. Thanks again for watching today. Thanks again for watching today, and good luck on your exams. I hope this helps. I'm going to try and do the protocols, but no promises. I'm low on time. So yeah, thanks.